Hello, my name is Emily Deruyter. I'm a senior ecology and environmental biology major, and I am presenting my honors undergraduate thesis. So my thesis was entitled Instrumental Evaluation of Residual Decomposition Odor Volatiles. And our question for my thesis was if the decomposition contact time with soil had an impact on the amount of volatile organic compounds that are found in the soil. And we were wondering this because there is limited scientific research into how residual decomposition odor compounds change over time, with even less research into how the compounds change as a function of contact time between the decomposing substrate and the decomposition environment. So the purpose of this study was to monitor abundances of the target odor vapors that were emitting from the soil after removal of the decomposing human analog. Human remains leave odor traces in the soil during decomposition, and they typically degrade over time. And it is hypothesized within our study that the longer the remains are in contact with the soil, the greater the number of decomposition related VOCs will be detected within the headspace of the soil sample. This should also suggest that there is a associated greater human remain detection canine response to the graves the longer the analogs are allowed to decompose in contact with the soil matrix. Since there is little research in the specific area of forensic taphonomy and none performed in the Colombian environment, the results of this experiment will provide novel data that can help in the detection of clandestine graves. So this experiment took place in collaboration between the Institute for Forensic Science at Texas Tech and the Colombian National Police at the Colombian National Police Force Canine Training Facility in Bogota, Colombia. We had 4 sampling zones, and each sampling zone consisted of 3 experimental sites and a control. Um, each sampling zone corresponded with a decomposition time that we were interested in. So we were interested in examining decomposition after 24, 48, and 72 hours, and then additionally 90 days. After the contact period that we were interested in, the pig would be removed from the soil, and then the soil sample would be taken and placed in a 10 milliliter vial that was then shipped to the Texas Tech Institute for Forensic Science. Um, we used a total of 12 pigs for this study. Each zone or time had 3 replicates and a control. This ensured that we had limited confounding factors and that we were able to understand what the background volatile organic compounds were and allowed us to identify specific to decomposition. So once we received the soil samples, we placed them in the freezer. Once we were ready to test them, we removed them from the freezer and allowed them to equilibrate for a period of 24 hours. After this 24-hour equilibration period, we then allowed a 24-hour extraction period. The extraction period took place on a hot plate at 40 degrees Celsius, which enhanced the vapor concentration within the headspace. Extraction was done by placing a spumy fiber within the headspace of the sample and allowed the VOCs to collect on the spumy fiber. After the extraction period, the spumy fiber was removed from the soil sample and it was injected into a GCMS system. The GCMS system allows us to identify the specific compounds on the spumy fiber and within, therefore, the soil sample. So the GCMS system operated for a total of 30 minutes, and it started at 40 degrees Celsius and ended at 250 degrees Celsius. And this was following the literature on the expected extraction processes for this soil sample. Our experiment found 7,353 VOCs at a quality match of 90% or greater. These compounds were tested over a period of 9 months due to the COVID-19 pandemic. Of these compounds, there were 16 classes of organic compounds that were identified. And this consisted of alkenes, ketones, nitriles, ethers, and esters. And finding these compounds was consistent with the literature on decomposing human analogs. Within the 7,353 compounds that were detected, aromatic and alkane compounds had the highest area within the sample. They're most prevalent within the experiment. And while this was consistent with the literature, aromatic compounds are typically detected within advanced decay. And we found aromatic compounds just after 24 hours of decomposition. And that is not a trend that is typically seen. And so we believe that this is due to the increased vegetation within the sampling sites. We also found sulfur containing compounds, and this was very important because they are indicators of decomposition found within 12 different decomposition analysis experiments. As I previously stated, we believed that the vegetation within the experimental sites may have impacted the amount of aromatic compounds that we were able to detect. And this is important to understand because vegetation can impact the accuracy of human remain detection canines alerting to decomposition. This is because aromatic compounds are typically seen within advanced decay, but they're also found within vegetation. So if there's a highly vegetated area, this might confound the canine and prevent it from detecting decomposition. So we were interested in examining two different vegetated areas. And so we split our zones into highly vegetated and sparsely vegetated. Um, this would be zone 1 and 2 are less vegetated, and then zones 3 and 4. We were able to do this within a graph, and we saw in the graph that the average area of the soil sample from almost all the highly vegetated areas had a higher composition of aromatic compounds. The high vegetation also seems to be associated with higher abundances of alkanes and sulfur compounds as well. This indicates that human detection or human remain detection canines might have a more difficult time alerting to decomposition within a more heavily vegetated area. This might cause false identifications of remain sites. So, understanding the chemical components of residual odors can allow forensic investigators in Colombia to improve their search and recovery techniques. The findings of this research determined that the vegetation levels surrounding a decomposition site can directly affect the aromatic volatile concentration levels in the soil samples. And so this would indicate that human remain detection canines might also have a harder time differentiating between compounds that are associated with death and compounds associated with vegetation. This information also has the potential to be used in criminal investigations to provide evidence about the transfer of cadavers from their initial burial site. Additionally, our research indicated that sulfur was not detected in the soil after 72 hours of decomposition. These findings are novel and can provide clarification to the compounds left in soil as a function of decomposition contact time. We should do further research into how this might affect human remain detection canines and their assessment of areas. Um, we should also examine the effects that the extended freezing due to the COVID-19 pandemic might have had on our samples. Thank you for listening to my thesis. I hope you all really enjoyed it. I had a wonderful time. So thank you to everyone that helped me through this process, Dr. Tiedemann, Dr. Canascaro, and Dr. Hoods.